Good morning, everybody. I'd like to thank everyone for joining us for our W.E.S.T webinar today, Wednesday, March 31st. Today we have our guest speaker, Dr. Christian Boada from Texas A&M University. He'll be presenting on biomimetic nanoparticles for the cardiovascular disease. As always, I wanted to first introduce our guest. Our guest here, of course, is Christian Boada. He's a man of the Americas, shall we say, from Ecuador all the way up to Michigan. I'm sure at some point he'll be out there visiting Canada as well. He could say he covered from north to south of the Americas. A little bit about Christian. He's gone to a lot of different academic institutions as well, across not only the U.S., but also in Mexico, down there in the Tecnológico de Monterrey, where he got his BS and his PhD. He's currently at Texas A&M, as I mentioned, but he's also been there at the HMRI and at Duke University. I'd like to also introduce the W.E.S.T team, which you're very familiar with, most of us, if you've been joining us for our journal clubs or these W.E.S.T webinars that we have. I'd like to start off by introducing our Southwest team, beginning with Viet Nguyen. He's our field application scientist down there in the Southwest, covers from Canada, pop in there, Kim, sorry. From San Diego all the way out to a little bit west of the Mississippi. Our regional sales manager, Kim Killian, partnered up with Viet in the Southwest. In the Northwest, we have, of course, Ian Villamagna. Ian will be the guest host, I guess, or manning the questions, and I'll talk about that in a little bit. And of course, I am Angel Galarza. I am the regional sales director for the W.E.S.T team. And welcome to our webinar. And last but not least, before we start, I wanted just to give you a little sneak preview of our upcoming webinars for the second quarter, beginning with Dr. Gaurav Sahai in April. Of course, then we have Dr. John Lewis in May, and we have Dr. Rickner on June 30th. As you recall, these are all on the last Wednesdays of every month. So if you can't remember the dates, just remember it's the last Wednesday of the month, 9 a.m. Pacific, 11 there in the Central Time Zone. W.E.S.T webinar, of course, now starts for webinars empowering science and technology. We got a little creative there. And of course, don't forget to check out our YouTube playlist. You can search them by W.E.S.T webinar and they will pop up. Without further ado, I'd like to introduce, of course, Dr. Christian Boada. Christian, the table is yours. And welcome. Welcome, Christian.
All right. Thank you so much. It's a pleasure and an honor speaking today. And truly have to say that Precision Nanosystems quite literally made my PhD possible. I will be talking on some of that today. As we have said, the title of the talk is Biomimetic Nanoparticles for Cardiovascular Disease. And in a way, I kind of jokingly include that or how I learned to stop worrying and love the nanoparticles, because it was quite a wild ride, but I'm very glad of the work we did. Again, Precision Nanosystems has been great. I've used their machines to kind of pioneer this technology, and looking forward to what I can do next. The principle here, so one of the things that was taught to me when I was in graduate school is, and this is all thanks to Dr. Anya Tashiati and his team, is the concept of biomimicry. And the way they incorporated that was basically they took nanoparticles and included the membrane proteins on the surface. And that is the principle, is literally to take the cell functions that you have on available to you and literally transfer them to your lipid nanoparticle. And the great thing about this is that, one, it's flexible enough that you can kind of tailor your application, and two, it retains all the qualities that we kind of know and love about lipid nanoparticles in particular. It's a great delivery vehicle, it's biocompatible, and now with this, you kind of have this increased and more sophisticated kind of tool set to work with. And you can see here a schematic of the nanoparticle. You can see the in red, you can see a little bit of the lipids, and this is a mix of the synthetic phospholipid that we add and a natural phospholipid that is already present either within the membrane or adhere to the proteins themselves. And obviously, you can see here that a variety of proteins can be functionalized on the surface. And the start of the show for this is really one of the things that kind of enables that technology is the precision nanosystem kind of microfluidic system. The way that we kind of go about making these biomimetic nanoparticles is that we take surface proteins, purify them, in a way extract them. We solve them in the aqueous phase, and then we have in the lipids, you had organic phase. They combine together in the microfluidic system, and you can create these biomimetic nanoparticles. And it does enable, because it's very convenient, more so than thin layer extrusion. And you can see here the characterization. You have nanoparticles that can vary in size, but for example, they're roughly around 100 nanometers, 122 nanometers. PDI, they're very homogeneous. And then you can see here the variety of proteins that can be functionalized on the surface, from transport proteins, from signaling proteins, immunity, adhesion. So it's really kind of a logarithmic increase in the number of things that we can have on the surface. And this is truly what enables it to have kind of these more sophisticated properties that, frankly, we're still finding out. We know there's a lot, it helps a lot with targeting, but we've also found that there's some implications for inflammation, for tolerance, and for other things that, again, we're just kind of trying to still figure out and still trying to harness this power and see what the applications are. Like I mentioned before, the great thing about these particles that we call the leukozones is, one, the synthesis procedure is very, very versatile. If you can switch the protein source, it is possible to create all sorts of matter of particles tailored to the application. If you're looking at white blood cells and you're looking to target something or a disease that you have accumulation of white blood cells, then you can use that as a source. If you're looking towards the central nervous system, then you can have cells that are related to that organ, and even combine to create new kinds of chimeric nanoparticles. The protein source, again, is only limited to purification procedures. You can have standard cell lines, you can have primary cell lines, you can have things derived from blood or plasma, or even recombinant proteins. It's really kind of a very diverse tool set, and the application is really limited by your imagination in this sense. The great thing about it is also this is still a drug delivery vehicle, and you can have various payloads. You can have small molecules, you can have genetic materials, and you can also have proteins encapsulated on the inside. And the design workflow for this system is kind of, we look at target disease mechanism. So we begin looking at the pathophysiology of an actual disease before we even make anything. Then we kind of synthesize and optimize, and then we kind of go forward and test in small animals. So it's something that can so it's not an isolated procedure. It's something that we hope can be taken out by other laboratories and kind of be expanded upon. So for my particular application, it was like, okay, so we have these nanoparticles, they target great, what do we do? And one of the first things that we have is, at the time I worked at Houston Methodist, well, I still work at Houston Methodist, and we have a great center for cardiology, and there's a great kind of tradition with the BACI, with the innovation for the LVAD, and so there's a great history of cardiovascular disease. So one of the questions that came up, why don't we just try to see if there's any solutions that we can come up with for cardiovascular disease? And one of the things that were kind of coming out of this is, the question is, why do cardiovascular disease, despite everything, and I think only maybe this year that has changed, is the number one cause of death worldwide? And the number two question is, how can we apply nanotechnology to solve this problem? So we began looking at the disease, and when you look at things like atherosclerosis, when you look at diabetes, when you look at all these things, the first thing you see is that at the beginning you have inflamed endothelium. And at least, for example, in atherosclerosis, the inflamed endothelium kind of shows these changes. Normally the endothelium is, I would say, very slick. You normally don't have any adhesion. Things just maybe roll onto the surface, but don't really stick. It is meant to keep the blood flow going smoothly. However, when you have this atherosclerosis and inflammatory procedures, the first thing that happens is this endothelium changes. And it changes, it becomes activated, it becomes inflamed. And what happens is at the surface, the phenotype changes to one of non-adhesion towards adhesion. So you start getting all this adhesion molecules that recruit lymphocytes, and then you have infiltration of recruit monocytes, and you have infiltration by the macrophages, and the macrophages drive that inflammation and further perpetuate that cycle of inflammation. And we saw this as an opportunity for localized delivery. If you can target these areas where the endothelium is inflamed and changing, that you can kind of shunt the disease and kind of steer it in another way. So we figured that this would be a very good approach to actually develop targeted therapeutics that would localize to targeted and to activated endothelium. And this had applications, like I said, for atherosclerosis, for diabetes, even for cancer. So we picked this as our target, and just looking at the facts of cardiovascular disease, it even make more sense to us because it's, again, 1 in 3 of all deaths in the US. And again, this may have changed this year, but it still generates, $351 billion in direct and indirect costs that is related to fatal events and the morbidity related to that. So loss of productivity, people having to stay out sick, loss in productivity as far as work goes. So it's a disease that actually costs more than cancers and all other infectious disease combined. So it's really something that has a desperate need to develop novel therapies. And as we began to look at the current limitations of cardiovascular treatment, we saw that despite being a wide variety of well-accepted kind of treatments for cardiovascular disease, the prevalence just really remains very high. And then the gold standard, which is cholesterol control, triglyceride control, is even in 20% of the people that reach that suggested cholesterol level, you still present thrombosis. And even now with COVID, we're starting to see people who are not normally at risk for heart attacks or thrombotic events. We're starting to see those people kind of having heart attacks all of a sudden. So we think it's very much related to the inflammation, and particularly to the inflamed endothelium at different sites throughout the vascular tree. And then the limitation number 3 that we saw is the guidelines for cardiovascular disease are mostly based on cholesterol and don't tend to recognize that much inflammatory component, although that's changing. So we are looking to kind of follow that rabbit hole and see where maybe treating the inflammation in cardiovascular disease is actually the better way to do that. What can we learn from the basic pathophysiology? Focusing in atherosclerosis, we saw that, just looking, it's a very localized disease. There's, on the available research, there's a strong correlation that sites of low shear stress correlate with sites of plaque development. And those shear stress manifest in kind of a lot of ways. Naturally at bifurcations, where the flow kind of changes direction very rapidly and very sudden. You can see there's areas where the blood flow kind of slows down a little bit. And so at any given stage, also you can see that there's development all over the vascular tree. And these are just the areas where they are commonly found. But at any given point, there may be different plaques, different stages of development. However, even though this is the case, the lesions are not diffuse, they're very well defined. So we're starting to see that there's a very highly localized disease. And the kind of obvious question is, how can we deliver the therapeutic directly to sites of plaque development? And again, therapeutics are very well characterized. In vitro, they are known to work. And then you see kind of discrepancy whether when we actually go into clinical trials, and there's been kind of puzzle over what's happening, and we do think it's because of targeting. If we're able to target this directly, I think we could be able to increase the available treatments by a couple order of magnitude just by virtue of kind of targeting these areas. How do we do that? The obvious questions. Working in a nanoparticle lab, well, the answer is nanoparticles, and we've published this. We've seen that the first iterations were kind of, we took membranes from different cell types and put it on the surface in a nanoparticle, and that worked, and that was very interesting, but it was still kind of cumbersome. Then the second iteration was the one that we see now, which is just a liposome with membrane proteins functionalized on the surface. And as we keep investigating, we see that these nanoparticles derived from white blood cells, monocytes, and activated monocytes are great kind of tools to get these target delivery over this activated endothelium. So the first project I'll be talking about today is one that I published in Circulation Research this year. It's called Rapamycin-Loaded Leukozomes Reverse Vascular Inflammation. So we are testing the efficacy of the leukozomes in the mouse model of atherosclerosis. And our hypothesis was that, like I said before, the increased accumulation of a drug, in this case rapamycin, would lead to an increase in efficacy. And that the local action of rapamycin will also reduce macrophage proliferation. And since the macrophages are kind of the linchpin that drives the inflammation cycle in atherosclerosis, we operate under the hypothesis that if we decrease the proliferation and action of these macrophages, we would reduce the vascular inflammation and therefore the growth and progression of the atherosclerotic plaque. And you can see a schematic here. You have the leukosomes, and within that leukosomes is the rapamycin, and you have the it acts upon the mTOR pathway, upregulating certain factors and then downregulating NF-kappa beta, which has a lot of downstream effects. It would have decreased adhesion, which is one of the drivers for accumulation of inflammatory cell types over atherosclerotic plaques. It would lead to decreased macrophage recruitment and also decreased inflammation. So the leukosomes in itself, we know that from other studies that we've done that they do localize very heavily to atherosclerotic plaques. And you can see proof of that here. We have some mice that were injected with liposomes, and that is bare lipid nanoparticles with no proteins on the surface, and our leucosome nanoparticles. And you can see on the left, the bright field image, the kind of whitish cloud is actually a plaque in mice. We take these mice and open them up, take the aorta, clean up, and then prepare it, open up the, the aorta so it lays flat. And you're able to actually visually see these lesions, and you can see that here. When we inject the liposomes labeled with rhodamine, you're able to see there's very kind of minimal accumulation over the plaque, maybe in a couple of areas here and here, but for the most part, it's just kind of not there. When we do that for the leukozome, you can see here in red, the accumulation is much more prominent. And when we actually measure that, it's actually a 4-fold increase in accumulation. So we're able to see that just compared to normal liposomes, you're able to get 4-fold increase towards atherosclerotic plaques. So we injected these rapamycin-loaded leukosomes into mice, took at the aortas, digested them, and then through cell sorting and flow cytometry, we looked at the percentage of proliferating macrophages within aortic tissue. And we had a control group, which was no treatment at all. They just developed the disease normally. We had a free rapamycin group, which we would figure in theory is kind of your standard to be. And then we had our particles with rapamycin. And you're able to see here that at least from these studies in 10 mice, the difference between free rapamycin and leukozome-loaded rapamycin is actually pretty significant, able to lower the proliferating macrophages within the aortic tissue. And we're not only able to see that, but we're also able to see kind of the downstream effects. We're able to see that the leukosomes also reduce some inflammatory markers. You can see here MCP1, which is macrophage colony proliferating factor, which kind of induces the proliferation of macrophages actually lowered, even when controlled with free rapamycin. ILB1 levels are low, both in free rapamycin and leukozome rapamycin, and C-reactive protein, which is kind of essential markers of cardiovascular risk in humans, is also much lower than the free rapamycin. This was a study that was done on mice that developed atherosclerotic disease, and then we treated these mice for 7 days. So we measured these markers, and we were able to see that there was a decrease right away, but we measured also plaque parameters, plaque physical parameters, to see if there was difference. However, not really expecting that to be the case. Against 7 days. This is a disease that develops over 12 weeks, so the chances of that happening was very slow. However, we did look at these nonetheless, and we were actually quite surprised to see that even after 7 days, there were some differences within the plaque percentage that we actually saw within this tissue, and directly correlated to that kind of the lumen area, which would kind of give you a measure of actual blood flow within that tissue. So we're able to see that leukozone rapamycin was actually able to improve that after such a short dosing schedule. And again, the conclusions from this was the suppressed macrophage proliferation in aortic tissue slowed the progression of atherosclerosis, kind of helping us make evident or establish the case for kind of targeting macrophages as a method of treatment of atherosclerosis. We were able to reduce the enzymatic activity, the reduce inflammatory cytokines and chemoattractant signals of macrophages, all with a favorable toxicity profile. And the result, even in the morphology plaque, was extremely surprising given the short dosing regimen of 7 days compared to the disease evolution, which is actually 12 weeks. And overall, this was very encouraging because this is kind of the first animal study that we did, taking these leukozomes and actually loading them with therapeutic and applying them in a cardiovascular model. So we're very excited to kind of work further on that and see where we can do maybe in larger animals or kind of down the road. So we're currently working on that. However, that's not all. During this, I've also developed kind of my own nanoparticle. The leukozome was something that was kind of developed originally Dr. Roberto Molinaro, and I just kind of applied that to the cardiovascular model. However, one of the things we wanted to try is, all right, we're saying that the leukozomes have, or the biomimetic nanoparticles can be designed from anything. Is that really the case? So we set out to do just that. And for the second part of this talk is I'm going to be talking about these ApoB100 biomimetic nanovesicles for atherosclerotic plaque targeting. And this is the design of a new nanoparticle using a biomimetic beta design. So a little bit about the morphology and classes of lipoproteins that we normally see. LDL, this is the lipoprotein classes in the small intestine, usually begin with the chylomicron, which you can see here is almost 1 micron in size. And then the chemical composition as it progresses throughout the body is kind of changed. And we get the VLDL, there's also ILDL, and then we get your normal LDL. And when people talk about this molecule, they talk about bad and good cholesterol. However, there is really not such the case. I'm sorry, these cholesterol don't really travel by themselves in circulation. They actually take the route where they're a complex molecule that includes cholesterol, but has a protein on the outside of your molecule that kind of directs the biological activity of this cholesterol. And as the protein component, and you can see here that it varies, the apolipoproteins, as they're called, that kind of change as they go through the body, and especially in LDL, what is known as the bad cholesterol, the primary component is apolipoprotein B. And what's interesting about this, like I said, this regulates kind of the biological activity, but and the biological activity is the delivery of cholesterol to different tissues. And as it is the precursor for a lot of hormones, cholesterol internalization and uptake are actually very extremely regulated. So once the internalization stops, that's when you get high serum cholesterol. And when you get kind of the development of plaques is when the internalization by these receptors that really don't have kind of a stop signal becomes more prevalent. And then you get accumulation of cholesterol and the inflammation that comes with it, and the normal evolution of atherosclerosis as we know it. So we decided to take this protein and see if we can formulate it into a drug delivery vehicle, essentially kind of hacking LDL. And what we did is we took blood serum, purified it using FPLC, which is fast protein liquid chromatography, which is just a size exclusion column. And we had the purified lipoproteins, and then we used the precision nanosystems to create these biomimetic, what we call them aposomes. And the novelty of the process was that, one, we're using FPLC purification that is not normally used beyond analytical methods, to replace ultracentrifugation, which is also very popular, but It's very time intensive. Meanwhile, the FPLC would take maybe 30 minutes to give us 1 ml of the LDL that we needed. And also, obviously, the microfluidic assembly method replaces the traditional extrusion methods to make nanoparticles, which, again, for me, was very, very fortunate because, again, normal extrusion is actually very time consuming. So this allowed me to kind of iterate quickly and get things done quickly. When we actually went to, and we did compare both methods, we did compare traditional thin layer extrusion to our microfluidic methods, and we measured kind of the rate of ApoB enrichment. The standard method refers to TLE, and the nanoassembler obviously to the microfluidic method. But when we were looking at this and we took the two fractions from FPLC that had the highest cholesterol content, and we compare them, we start to see that the standard method, the thin layer extrusion, really is not very well suited. You're not able to see kind of a homogeneous populations or anything of that sort. Meanwhile, on the nanoassembler side, you're able to see clearly populations that have been enriched with ApoB 100, and even in both, even though in this, for example, this particular fraction was not, it's only 41%, you're able to see that in this particular fraction, you're able to get the majority of the nanoparticles with ApoB 100. And just to kind of think about that, I think it's the thin layer extrusion method kind of subjects lipids and the nanoparticles to a lot of forces. So it's temperature, it's kind of the shear force that you put in a particle as you push through the membranes to get the size that you want. And with nanoassembler, with the microfluidic methods, that's not really the case. So I think that had to do something with it. And it would be a matter of, even if it wasn't for the microfluidics, I don't think we'd be able to make this using standard thin layer extrusion methods. When we look at the ApoB enrichment using other methods such as Western blot, we're able to see that the aposome, even though, and again, this is purified from from whole world plasma, you get a lot of proteins, but you're still able to see a very prominent signal for the ApoB 100. You can see where we compare it to pure LDL. It's very comparable. We also took a look at other lipoprotein classes that may be present, and one of the most prominent apolipoprotein classes was ApoA1. And we saw that that was not the case. It was not present. So Western blot obviously confirmed the presence of ApoB, but we also see that the FPLC method itself was a good method to get the majority of other apolipoproteins contaminants that would be within whole blood plasma. As we kind of looked at the aposome structure, you can see here the liposome using cryo-TM. You can see the liposome, circular structure, membrane double layer, the low density lipoproteins, very small, kind of this dense structure where you have the ApoB on the surface and then the cholesterol ethers on the core. And then you have the aposome that resembles a lot more the liposome structure. However, when we took a look at the thickness of the bilayer, we saw that the aposomes actually had a thicker bilayer, pointing out towards confirming that the ApoB was actually embedded on the surface. Membrane thickness on cryo-TM is usually an indication that there is protein, that there is protein functionalization on your nanoparticles. We further confirmed that using flow cytometry, looking at kind of antibodies that would bind to ApoB, and we're able to see that even after 48 hours, the aposomes retain their ApoB on their surface. So we were able to get stable synthesis methods that would have ApoB on the surface. And now it's just a matter of testing their safety profile. For this, we used some of our collaborators at U of H help us do this. We looked at capillary and red blood permeability as a marker of safety. Any changes that would happen to blood is a very extremely sensible indicator of activity that would be affecting the red blood cells. And if that happens, you have clotting, and then you have all these things that would kind of signify that they're having an effect, a deleterious effect towards the red blood cells. However, that was not really the case when we compare liposome to aposome. the profiles were pretty much the same, and the liposome was actually, and the liposome is a known drug delivery vehicle that is compatible and safe. So to see that there is no difference with the aposome was encouraging, saying that this has a very good safety profile. We also tested one of the questions that kept coming up. You're injecting this foreign protein into a body, and in the best case scenario, you can probably get it from the patient. But if not, what are the chances that you could develop a reaction to it? And that was actually a very good question that we tried to answer. We took human LDL and injected it into mice at a very high doses for 1 month. Then we took the plasma, purified it from those mice, and then incubated new liposomes and aposomes in that plasma to see if there are any antibodies that would bind to the surface. We checked IgG and IgM because those are the two kind of most prominent immunological Ig classes that would be binding to any kind of foreign material. And we saw that even after the treatment, there was really not that much increase in the antibody binding towards the apolipoproteins. And given that these are from two different species, one was mice, it was injected in mice, and the protein source was human, it was very encouraging, saying that there's no apparent immunogenicity, specific or unspecific, after prolonged exposure. And there's also no alteration of metabolic marker, which is not included here, but even LDH and common things like that, we weren't able to see any significant difference in the biocompatibility profile. So that was encouraging from a safety standpoint going forward. Now, we injected this aposome technology into mice that had developed atherosclerotic plaques to test their accumulation within these places. And you can see here a familiar image where you have the bright field, the cloudy image, and it corresponds to the mouse plaque that was developed 12 weeks after a high-fat diet. You see here in the fluorescence pictures, liposomes are on the left, aposomes are in the middle, and these are actually mice that were previous to injected with an CD68 antibody. And I'll talk about that in a little bit. But when you compare liposome to aposome, the difference is actually almost 20-fold. And in some cases, the difference was so extreme that you can see it dwarfed all the other experiences. But we were able to see an average of a 20-fold increase accumulation over plaque. And what happened was we were trying to see if this was colocalized to macrophages or maybe it was another type. So we injected with a CD68 antibody to kind of visualize the macrophages and see if there was any accumulation. And when we injected the aposomes again, there was no accumulation. And we repeated this a couple of times and we saw absolutely nothing, which is very strange because, again, you're able to see this profile that is just, there is extreme accumulation in these plaques. And this is a representative picture of kind of all the plaques that we saw. You're not only able to see aposomes kind of on the side, but you're also able to see them in the core. So we hypothesized, and we're still doing kind of in vitro testing on this, that it's probably a CD68 dependent mechanism that helps the internalization of the aposomes. And that's what we have so far. We show that the biomimetic nanoparticle that has ApoB on the surface can have a 20-fold increase targeting over atherosclerotic plaques. We have extremely favorable safety profile that shows really no alterations. The particle do not induce an immune response even after prolonged exposure. To our knowledge, is the first instance of such a large protein being functionalized in the lipid-based nanoparticle, especially using a microfluidic synthesis techniques. And the great thing about this is that the synthesis methods could potentially accommodate large-scale synthesis, overcoming a significant kind of problem with LDL-based therapeutics, and primarily ones that are derived directly from whole blood plasma. So that's very encouraging. Right now, what we're working on is testing these nanoparticles, obviously in larger animals, both the leukozome and the aposome. We are going to continue testing to add additional payloads. We can do hydrophobic compounds. We'll do hydrophobic, hydrophilic compounds, but special interest is obviously RNA. So we're actively working on those areas. And we're always looking for collaborators. So if there's anybody that has an RNA, a novel RNA that they want to test, and then maybe they see some utility for the nanoparticles, then it would be great to kind of get in touch and maybe talk about doing a collaboration together. And obviously, I will have to bring this up, the continuous simplifications of the synthesis procedures. As we get better with this, we want to make sure that these technologies are translational. So we're always working on new ways and better ways to kind of make these nanoparticles so that they have the best chance of kind of making it forward into a clinical therapy, into a drug therapy. So that's currently the direction of our lab. And I would like to thank you, everybody that's listening today, for taking the time to hear me talk. I would like to thank Roger Pettigrew, who is my mentor at Texas A&M, Dr. Cook, who is currently my mentor at Houston Methodist, my PhD mentor, which is Anita Shadi, Dr. Singer, and Dr. Jonathan Martinez, who helped me enormously through my graduate work, and my university and the Mexican government for supporting this work and allowing me to study this. So thank you very much.
Absolutely. Great job, Christian. I really appreciate it. Well, thanks again for joining us today. Not only Christian, of course, but everyone that made it and that still is around. Please remember that we do have our quarterly, not only of these W.E.S.T webinars that we have, but we also have journal clubs that are the second Friday of every month. And of course, we have a lunch and learn, which we're adding to the rotation because we do think that is important to continue to communicate. So look out for the invitations for not only our W.E.S.T webinars, but also our journal clubs, which is the second Friday of every month. And we also have a lunch and learn that we'll be having. Of course, it'll be virtual, so we do apologize for that. We hope to get those running back live. Thanks again. Please go to our website and do join and pre-sign up for the actual journal clubs, and also, of course, our W.E.S.T webinars. Christian, thank you very much. Everyone have a great day.